我们3:45开始啊，3:45。嗯。Test. Test. 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 Test.
你再买点彩票。现在运气这么好，我们看有，哎，上地方这个是好，这个是别的。
那我先开始，我先介绍你们先这个。OK， 好，那我们现在就开始了啊。I think I can speak both Chinese and English. OK， 那个我和王照，我们是这个 section 的chair 啊。我先介绍一下Dr. 赵王啊。Go with Jill. Dr. Jill Wang is an expert in the pharmaceutical industry with 70 years of experience focused on regulatory affairs. Dr. Wang is currently serving as a regulatory affairs director in CVRM at AstraZeneca. Jill has a PhD in pharmaceutical science and an MD in general medicine. Before joining AZ, he worked as both the FDA and Chinese FDA. So Jill has led a project in cell therapy, gene therapy for FDA and global regulatory strategy, and holds several patents for drug development.
Thank you, Dr. Han, for the nice introduction. Welcome all to this afternoon session. This session will focus to discuss on preclinical development from esteemed institute, the well-known professors, and also we have regulatory insight as well. It's my great honor to introduce our first speaker and my co-chair, Dr. Zhe Han. Dr. Han is a renowned geneticist known for using fruit fly to study human disease and develop new treatments. He earned his degree in genetics and development biology from Peking University and the University of Michigan, and did postdoc research at UT Southwestern. Dr. Han started his lab at Michigan, where he developed gene replacement technology to model patient-specific disease in flies. His work has helped advance research on rare cardiac, kidney, and cancer disease. Now at the University of Maryland, he leads the Center for Precision Medicine Modeling and has published around 90 papers with over 20 million NIH funding. Now it's my great honor to welcome Dr. Han. The floor is yours.
Thank you for the introduction, and I'm glad to have this opportunity to talk about our Drosophila modeling for precision disease model. And I'll give you some example on how to use Drosophila, which is a high throughput drug screening platform to tackle rare genetic disease, particularly in heart disease and kidney disease. Okay, so what is precision disease modeling? And precision disease modeling, there's no defined answer from the dictionary. So different people have different opinion on this. But through the center I established and in Maryland, we want to establish precision disease modeling like this. So what we mean by precision disease modeling is personalized disease modeling, in which we put the exact genetic variants identified in patient into the animal models. And we will have a relevant phenotype from the animal closely related to the human patient. And the goal of the precision disease modeling is to identify disease causal factors, discover a mechanism, and then develop mechanism-based targeted therapies. And we can also use a precision disease model to do drug testing and drug screening. So this is a very early preclinical step to develop treatment for disease that currently have no cure. So why use Drosophila? Because Drosophila has a surprisingly very high homology to human. So more than 60% of human gene have fly homolog, and 75% of human disease gene are conserved in fly, and most human tissue or organs have counterpart in flies. And Drosophila is actually the it sit at the very sweet spot among all the modeling system for disease because it is the most complex high throughput animal model. So it's much complex than those simple models like worm or yeast, but it still has most of the organs compared to mouse and zebrafish, which are more expensive and take longer to generate disease models. And studies in Drosophila has led to 6 times Nobel Prize, identify many conserved genetic factors and the principles in developmental biology and therapeutics pathways. And Drosophila, among all the model system, has a very powerful genetic tools. Oops. Okay, so in fly is actually the only model system where you can effectively remove any gene from any cell type at any given time. So only fly can achieve this. This is achieved by the US Gaithersburg system. Let's keep. By using the US Gaithersburg system, which I will tell you a little bit more in my next slides. And also the gene replacement approach, which developed from my lab as currently the most effective way to model human disease. So I'll give you some example. We'll start with the heart disease. There are 2 major types of heart disease: congenital heart disease, which is a developmental defect, and dilated cardiomyopathy, which affect both pediatric and adult. So the way we model the disease is to start with the patient, identify a particular gene and particular mutation, and then we will identify the fly homolog of the gene changed in the patient, and then we can easily silence that gene in the relevant organ. In this case, we remove that gene from the fly heart. If the gene is important for the heart development or function, will have a phenotype. So this is what we call the gene level validation. Based on the gene level validation, we also can go further to do the variant level validation, in which we put back the wild type human gene in the fly heart, use the human gene to replace the function of the fly gene. And in parallel, we put the patient variant allele into the fly heart. And then we compare the wild type human gene versus the patient derived variant allele to see which one can rescue the defect better. If the variant really affect the human gene function in the heart, then the wild type human gene will rescue better. And then we have the functional validation of the variant using Drosophila heart. So this is a principle of the gene replacement approach we developed. So the reason we can use this system to model congenital heart disease is because the heart development is highly conserved from fly to human. So this is a lot of study from my lab and other lab has established the genetic regulation of heart development and the conserved transcription factor and pathways. And my lab is the first one to use Drosophila heart to validate 138 candidate congenital heart disease gene and to identify which gene has conserved the role in heart development from fly to human. We're also the first lab to establish what we call the precision disease model for a particular congenital heart disease patient. So we put the patient allele back to the into the fly heart, replacing the fly homologous gene, and establish the functional requirement for that variant. We also generated the dilated cardiomyopathy fly model. In this case, we removed the gene, a fly gene, which is a homolog of the human LMNA, a very famous dilated cardiomyopathy gene. Removing of the LAMC, which is a fly homolog of LMNA, gave you a dilated heart, a typical phenotype to dilated cardiomyopathy. And in this background, we put back the human LMNA, we can rescue the phenotype. Therefore, we have a platform to put in all kinds of genetic variants that affecting LMNA. So currently, there's thousands of LMNA variants identified from sequencing. Many of them, we do not know whether or not they lead to disease. So this is a highly effective platform to test those variants for their function, and also establish what we call the precision disease models. And in the future, we're going to use these models to test potential therapeutics. And this is the variants that we already tested in the fly heart. Using the functional data, we can establish whether or not those are disease-causing variants. Another example is the fly kidney, which my lab is a pioneering lab to establish Drosophila kidney disease model. The principle is the same, the gene replacement. We start with the patient. We identify a particular human gene with a variance, maybe causing the kidney disease. We identify the fly homolog, we silence it in the fly kidney cells, and we see a kidney phenotype. In this background, we put back the wild-type human gene and the patient variant, and to see how whether the wild-type human gene can rescue better than the patient variant allele. If it rescue better, then we have the much needed functional data to show that the disease is indeed caused by the particular variants. So we have established more than a dozen models in the past 10 years. So this is to show you the fly renal system versus the human renal system. And my lab established the first in vivo functional readout for kidney function. Here, if you see the Drosophila kidney cell, which we call nephrocyte, if it turns right, that means it has function. If it's no longer right, that means it doesn't have any functions. So it's very easy to test readout. So using this readout, we went through all the known genetic kidney disease called nephrotic syndrome, which is a particular severe kidney disease that no cure has been available until recently. So we found that 85% of known nephrotic syndrome genes have fly homologs required for the fly kidney cell nephrocyte function. And we using this platform collaborating with a clinician, we identified the genetic causes of a previously unknown kidney disease family, and we identified the gene is NUP160 as a novel kidney disease gene. So if we silence the fly homolog of NUP160, the kidney cell completely lost function. You see, there's no red fluorescent signal. And when we put back the wild-type human NUP160, the kidney function is rescued. And when we put the parental allele from father, from mother, back into the fly kidney cell, the function does not get rescued. Therefore, we show that those variants are causing kidney disease. In another example, we not only made a precision disease model for another patient. This patient is from Italy. We also identified a potential therapeutics because in this case, the gene affected is called COQ2, which is a rate-limiting enzyme for coenzyme Q10 synthesis. And then we tested by a potential drug. Actually, this is a nutrient supplement called coenzyme Q10, which is actually the top 5 best-selling nutrition in the United States. So we feed the fly model coenzyme Q10, and it rescues the renal defect. So this is actually the first case that using fly model, using fly kidney disease model, identify a mechanism-based treatment, and then right now this coenzyme Q10 treatment has been applied to patient. So making the COQ2 associated nephrotic syndrome the only type of disease which is currently treatable. So all the other nephrotic syndrome are still not treatable, and we're working on it. So I'll give you one more example on how the powerful fly system can be used to do drug screen. In this case, we modeled leukemia, particularly is a KRAS G12V induced acute myeloid leukemia. In this case, we introduced this very strong leukemia causing variants in the human gene called KRAS, KRAS G12V into the fly blood. And because of this leukemia variant, the fly blood increases number over 700-fold. So you can see that the entire fly right now glow with the green fluorescent protein, which labels the blood cell. So this is a great model to do drug screen. So we use this model and we feed the fly all kinds of drug, and we identified at least 2 drug candidates that can inhibit this leukemia phenotype. Just by this is also very easy to score. You just look at the fly and you see the green fluorescent signal is reduced. That means the drug worked. It reduced the leukemia, the number of the leukemia cell. At the same time, we also use this leukemia model to do genetic screen, and we identified the hypoxia pathway gene, including the SEMA and TANGO, when silenced, also rescues the phenotype. Together with this drug screen candidate, we established that equinomycin, which is an inhibitor for HIF1 alpha, in fly the SEMA, can rescue the phenotype. So not only we identified the drug, we also identified the mechanism of how to block this KRAS G12V induced acute myeloid leukemia phenotype. Okay, so we are now expanding this platform to target thousands of rare genetic disease. So there are more than 7,000 type of rare genetic disease has been identified. 95% of them does not have a treatment because the using the traditional way to develop treatment for rare disease is a very high cost and take a long time. So we try to use Drosophila as a high throughput platform to develop new treatment for those rare genetic disease. And the most of the genetic disease, rare disease, is caused by genetic variants, genetic change, and which can be identified by genetic testing. And because the sequencing cost is dropping dramatically, and the number of the variants with uncertain significance rise so rapidly, they prevent a tremendous opportunity to use our platform to make models for like hundreds of thousands of genetic variants that cause rare disease. So this is the reason that I established this new Center for Precision Disease Modeling at University of Maryland School of Medicine. So our center allows the clinician to submit the variants identified from the patient. So our center will do a free evaluation on how the possibility of the variants causing the disease, and then if it's feasible to model in fly, we'll make a fly model. If it's feasible for the fish, we can also make a zebrafish model. We also have a zebrafish facility. And using those models, we'll study mechanism, and then we'll develop a mechanism-based therapeutics. Okay, so that is my talk. This is my lab, and we're well supported by the NIH, and thank you very much.
Thank you, Dr. Han, for the nice presentation. Now, I think you will introduce the next speaker.
Okay. Okay, so it's my pleasure to introduce Dr. Hongbin Wang as our next speaker. Dr. Wang is a professor and interim chair at Department of Pharmaceutical Science in Maryland School of Pharmacy. Dr. Wang received his PhD in toxicology from Fudan University and did postdoc training at the University of North Carolina. Dr. Wang's research is focused on nuclear receptor-mediated regulation of metabolizing enzymes and drug transporters. Dr. Wang's research received continued support from NIH and FDA, has authored over 130 publications and served as reviewers for NIH and the FDA. Thank you.
Thank you. Thank you, Zion, for the nice introduction. And it's a pleasure to present some of our research. As mentioned, my lab mostly focus on using nuclear receptor and study the drug metabolism process and how this will affect the drug safety and efficacy. And this is one example. We developed a dual activator of two nuclear receptors, and it seems like this may benefit the traditional chemotherapeutic drugs, the cyclophosphamide and the doxorubicin. So first, I want to convince you why we are still interested in traditional chemo. And even with the rising wave of the immune therapy and the targeted treatment and also personalized medicine, the traditional chemotherapy still play a pivotal role in the overall cancer treatment. This is one study. They analyzed the global cancer incidence and the patient required chemo treatment. They used the 2018 data to estimate the 2040 incidence. And as shown here, you can see in 2018 globally there are about 17 million cancer incidents, and by 2040 there will be 26 million. And actually in 2018, from this 17 million, and there are close to 10 million patients still require traditional chemo treatment. And that number will be increased to 15 million. So you can see with the near future, there's the traditional chemo is not going to anywhere. However, the chemotherapeutic has been notoriously associated with severe side toxicity and also drug resistance. So how to deal with this situation and how to improve the efficacy and reduce toxicity? So we use two examples, and one is cyclophosphamide, one is the doxorubicin. These two drugs, they are quite often co-used for multiple clinical use regimens. And they are very frequently used in various different types of cancers, especially for triple-negative breast cancer, lymphoma, and leukemia. So the cyclophosphamide itself is a prodrug. So the drug itself will not kill cancer cells. However, it relies on the liver metabolism to convert the cyclophosphamide into the 4-hydroxycyclophosphamide, followed by several steps of spontaneous response, eventually generate this phosphorus. I think I should do something. So this okay. So eventually generate this like cytotoxic mustard, and that will target the cancer cell DNA. And so the liver conversion of or activation of cyclophosphamide is heavily relies on this cytochrome P450 called CYP2B6. However, the 2B6 has a very low basal expression in the liver, and also show large individual variations. So the transcriptional expression of the CYP2B6 is mediated by a nuclear receptor called constitutive androstenedione receptor, or abbreviated as CAR. And so activation of CAR can dramatically increase the 2B6 expression that favor the bioactivation of cyclophosphamide. And we would think about if we can have a small molecule can activate this receptor, whether that will favor the bioactivation of cyclophosphamide. So in the case of doxorubicin, this is another traditionally used chemo drug. A very severe and dose-limiting side effects of doxorubicin is cardiotoxicity. And many patients, they cannot continue the chemo, dox-based chemo, because it's lifelong accumulated heart toxicity. Okay, so mechanistically, doxorubicin causes the heart cell toxicity mostly by overproduction of reactive oxygen species and also induced mitochondrial damage. And we know the nuclear receptor NRF2 mediate a global or cell, antioxidant program. And so we'll think about if we can specifically activate the NRF2 in the cardio, cells, whether that can detoxify the doxorubicin-induced cardiotoxicity. So based on these ideas, we start to develop a dual activator of these two nuclear receptors. So we expect this dual activator will, on one side, increase the I'd better not touch it. I didn't. Yeah, you see. It is. Okay, so on one side, it will activate the CAR and increase the cyclophosphamide bioactivation and increase the chemotherapeutic effects. On the other side, it will activate NRF2 and protect the heart from DOX-induced toxicity. So we started with a high-throughput screening in collaboration with NCAT, and they have multiple chemical libraries. And we started with the CAR activation assay, and the selected CAR activators will further move on to the NRF2 activation experiment. And from there, we eventually identified the CN06 as the most effective and potent dual activator of these two nuclear receptors. Then we evaluated the function of this drug candidate by looking at their induction of 2B6 and activation of a CAR in human primary hepatocyte. As shown here, you can see the CN06 can dose-dependently increase the expression of 2B6 from mRNA to protein levels. And also down here, you can see the CN06, this compound can also translocate human CAR from the inactive cytosol located CAR, the yellow color, and move into the nuclear, and that indicates the induction of the 2B6 is mediated by the CAR activation. And next, we use an oversimplified co-culture model. And so at the bottom, you can see we have a human primary hepatocyte to mimic the liver. And in another chamber, we have target cell. Here's the triple negative breast cancer, and they share the medium. And so then we treated with the CN06 in a different concentration of, combined with different concentration of cyclophosphamide. As shown here on the left panel, you can see the BT549, that's a triple negative breast cancer cell line. You can see in the presence of CN06, the cancer cell viability is dramatically decreased compared to the cyclophosphamide alone. And this change is also associated with the right side. You can see, when we include CN06, there's an increased synthesis or formation of the 4-hydroxycyclophosphamide, which is the first and rate-limiting step of the cyclophosphamide activation. So next, we want to look at whether CN06 will be able to activate nerve 2, mostly in the cardiomyocyte. As shown here, we use the H9C2 cell line. That's a red cardiomyocyte cell line. And as shown here, you can see on the top panels, the treatment of CN06 can cause the nuclear translocation of nerve 2. That's one indicator of the activation. And we also look at the HO1, that's a hemooxygenase 1, that's a typical nerve 2 target gene. And you can see CN06 can increase that expression as well. Interestingly, this not happen in the cancer cell line, which means like you can protect the cardiomyocyte, but most likely will not, compromise the drug treatment effects in the cancer cell. And indeed, we use the iPS cell-derived human cardiomyocyte. As shown on the left side, you can see the drug CN06 itself doesn't cause toxicity, but the doxorubicin caused the toxicity can be dose-dependently rescued by the presence of CN06, and there's a functional experiment down here. They also show the similar protect effects. And very interestingly, and on the right side and the two cancer cell lines, their anti-cancer effects of doxorubicin is not affected by this new compound. So this selective protection is very important. And with that, we further modified the co-culture model by including not only the hepatocyte and the target cancer cell, we also include the key off-target cardiomyocyte, all share the same culture medium. And as shown here, and when we co-treat the co-culture models, and then you can see on the left side, and the presence of CN06 really enhanced the anti-cancer activity of this doxorubicin cyclophosphamide combination. And simultaneously, it also protects the cardiomyocyte on the right side. And that indicates this, this is a proof of concept, indication this compound may benefit the chemo treatment. However, this CN06 has proof of concept, information. But when we look at the chemical structure of this compound, it may associate with some toxicity. So in our department, we have expert, Dr. Fengtian Xue. He helped us to optimize this compound, design the next generation of analog. And this, preliminary data showed the new compound, the YA compound, compared to CN06. You can see here, it has a better induction of 2V6, better induction of the NERV2 target gene HO1. And also, when we look at the toxic profile, the blue color is the CN06 and the red color is the YA compound, and there's a much improved profile. So then we also tested this one in a patient-derived organoid system co-culture, and then similarly, we can see the presence of the YA compound can enhance cyclophosphamide-induced anti-cancer activity. So with that, I reached this brief conclusion. And so by this combined effort, we have identified a number of CN06 analogs. They are tissue-specific dual activator of these two nuclear receptors. When they are co-used with the cyclophosphamide and doxorubicin in our co-culture models, it seems like they can improve the therapeutic efficacy and lower the cardiotoxicity simultaneously. So most of the work is conducted by grad student Sydney Stern, and then she's now working at FDA, and then also collaborators. And thanks for the funding support. With that, I'll be happy to answer any questions later. Thank you.
Okay, thank you for the wonderful talk. I'll do a brief introduction for our next speaker, Dr. Yuntao Wu. Dr. Wu is a professor and co-director for Center for Infectious Disease Research at George Mason University in Virginia. Dr. Wu received his PhD in molecular virology from Queen's University in Ontario, Canada. He did his postdoc training at the NIH. Dr. Wu has over 35 years of experience in virology, focusing on HIV research. His lab discovered the roles of cofilin in HIV infection and pathogenesis. So in addition, for HIV host interaction, Dr. Wu is also interested in developing new technologies for HIV cure. Dr. Wu is a recipient of multiple NIH grants and served as the editor-in-chief of current HIV research. And he's also a frequent grant reviewer of the NIH and numerous scientific journals. Hey, yes.
Thank you for the introduction. Also, I would like to thank the organizer for give us this opportunity to share our research. So my lab study HIV infection. So today, so I like to share some of our recent data for looking for cure of HIV infection. So we use a different approach to targeting viral reservoir. So, okay. Oh, it's not working. It's not. The slides is.
Sorry for a little bit technical issue. We're trying to fix it. Just once more to remind the audience online and on site, we'll have the QA session at the end of this session. So please hold your question to that end. Are we getting better? Okay, good.
So this is the HIV the AIDS pandemic started in 1981. At the beginning, we thought it was a gay disease. A lot of infected people are gay people. Later on, we know 1983 the virus was discovered. It's not a gay disease. It infects everyone. So the virus is a retrovirus. Oh, sorry, the slides. Okay. Okay. No, it's not working. Okay, so. Okay, so. So we this was is a retrovirus. Really the virus, infect human T cells. The CD4 T cells, it get into helper T cells, CD4 helper T cells, and then it's a retrovirus RNA is reverse transcribed into DNA. So the DNA oh, okay, stop. So yeah, so I think he's controlling there. Yeah. Yeah, oh, not maybe. Okay. Okay. So I apologize for all the technical issue. So the genome, the viral genome DNA is integrated into human genome. So that integration allows the virus to stay in human genome essentially forever. As long as the cell is alive, the virus is there. So the discovery of HIV virus really opened the two doors. The first one is treatment. You can use antiretroviral therapy for treat patient. Secondly, really prevention, right? So we can do blood test and to identify virus. Also, we can develop vaccine to prevent infection. So the virus, immediately after the virus was identified, the AZT from NCI was immediately used to treat HIV infection. So this is drug, the first anti-HIV drug, AZT, and later on there's more drug was developed, right? There's one famous Chinese scientist, David Ho, he really took all the drugs together, 3 or 4 drugs together. We call it antiviral combination. End up it's fairly potent. So this is a paper published by David. So it only takes about 2 weeks. You can reduce 99% of virus, and almost few months is undetectable. The David is now is 1997. He was named as the man by Times Magazine as the man of the year because his mathematical modeling predicted if you use the called HAART, HAART, and highly active antiretroviral therapy, you treat it for about 3 years, you should cure HIV. So that's a prediction. But now we know, you can treat a patient for 40 years, 30 years. You can treat it for a long time. But once you start the therapy, the virus coming back right away. So the reason it was confirmed by multiple study fairly soon after the prediction. This is an early paper, 1997. It showed HIV coming back once you stop the therapy. This was treated for a few years. And also, this another study show even you treat a patient for 60 years, and then the virus, you may be able to stop the virus from coming back. So 60 years, that means essentially there's no cure, right? You need to treat the patient for 60 years. So we know now we know what's the reason, right? The reason is because there's a those are after 40 years we need to end AIDS, HIV/AIDS pandemic. Also, we need a vaccine. So far, we don't have a cure. We don't have a vaccine. So that the reason is because this virus, as I mentioned, is a lot of challenges for developing a cure as well as the vaccine. The vaccine is a lack of immune correlation with protection. We know COVID infect us, right? You recover, the virus is gone. There's no, we know almost nobody recover from HIV infection. So once you got infected, it's lifetime infection. The virus mutate a lot. Your immune system is not able to really generate good antibody to broadly neutralize all the mutants. So this also the virus reservoir persisting in the body. There are 2 types of reservoirs. It's a major reservoir. There are other cells as well. So CD4 T cells, the helper cells itself, carry HIV. The genome inside the virus, inside the cells, chromatin, also macrophage. Those are HIV infect cells. They persist in the body, especially the memory CD4 T cells in your system for many years. So now the HIV field is looking for how to clear this latently infected reservoir cells. They use an approach called shock and kill because the virus hiding in the cells. Can we activate the cells, shock them, so then they make them produce viruses? In the meantime, you use drug to block viral replication, in hoping that the reservoir cell will be cleared, will die either by infection or by your immune system. So you can call it shock and kill. You can end up wipe out HIV completely from your system. So those are approach. So far, also there's no indication that approach can work. So many labs tried all the, use HDAC inhibitor to active reservoir, but we don't have evidence to show it really can completely wipe out the reservoir, right? So my lab take a different approach, right? So instead of the shock and the kill, we are engineer. We want to use HIV to against the HIV. We took the HIV virus to engineer the virus into a HIV-like a particle. Essentially, take the genome, you delete all the viral genes, then only leave one few very simple element called RE, called responsive element, which is a trigger. I will go to detail later on. You make sure this particle can express gene only in HIV cells, right? You can deliver that particle as a Trojan horse into HIV cells. They're looking for HIV. If there's HIV, there's gene expression. So that's a rationale. So you can see the traditional way of treating HIV use ART, the antiretroviral therapy. It only kills virus. It does not wipe the reservoir cells. That's the reason you need lifetime treatment, daily ART treatment, right? So to keep the virus in control, right? You only kill the virus, does not kill the cell. So our approach is essentially use HIV-like dependent vector. This is a particle very much like HIV. We're looking for the reservoir cells. Then we either induce the killing of these cells or because another way we can inactivate them, allow them to only release defective particle. Even they release virus, the virus is a dead virus. Or you can use to stimulate immunity, to stimulate your immune system to generate antibody against the virus itself. In some extent, this Rab-dependent particle, they're the pest of HIV. You use the pest of HIV against HIV. So this is so we're using this, essentially engineer HIV to against HIV. Because it's Rab dependent, it depends on HIV for gene expression. They're high specificity. So also you can stimulate immune system. So this is the so basically we know that in the human system there's a small percentage of people we call the elite controller is about 0.2 to 0.5%. This patient can control HIV even without any treatment, right? They can control for many years. So we are thinking, can we use our technology to turn this 0.2% into around 50% patient, right? So you can control HIV, require your use your own immune system, use our particle to stimulate that kind of immunity, and the majority of the HIV patient can self-control without rely on drug. So that's a goal we try to achieve. Right? We can reduce the reservoir in the meantime, stimulate the immunity. So this is I mentioned, it's a Rab-dependent viral vector. The only thing in there is called RE, called Rab-dependent element. So it's a trigger. So HIV express a protein called Rab. If there's Rab in the cell, it binds to RE, it triggers gene expression. It's a fairly simple concept. So those are how the vector is constructed. So now I can show you. We can assemble HIV particle in the laboratory. That particle packaging genome have the RE, have the GIP, green fluorescent protein. We use this is as a reporter. We can show and do a proof of concept study to show is HIV dependent infection. You can see here, this is a HIV macrophage, right? Was in fact one of the cell have HIV, then we super infect the cell with our Rab dependent particle, and you turn off the light. There's one cell glowing. You can see that's HIV cell, right? It's only express that protein. The green fluorescent protein in the HIV cell, not in the healthy cell. You can do this and use flow to marking T cells. You can mark in large number of HIV infected T cells. It does not express GIP in healthy cell, only in HIV infected cells. So those are selective expression of gene in HIV positive cells. So we for therapy, you just replace a reporter like GAP. You replace that gene with a therapeutic gene, right? And then you can express that therapeutic gene only in HIV cells. So what we use here is a herpes TK gene, thymidine kinase. Then we can use a drug called Cancr to kill. So then we turn HIV infected cell into a herpes infected cell, like a cell. Then we can use Cancr to kill. So this is the mechanism of how Cancr is a clinical drug treating herpes infection. So we turn HIV infected cell into a herpes infected cell, then use Cancr to kill. So those are all this is a recent monkey trial. We use this approach. You know, the monkey has a HIV is very much like HIV. So we can do treatment, then stop therapy. And we inject the monkeys with the vector. I will just briefly tell the results. And the monkeys were treated with one time of the particle. You can have about 100 to 1,000 log decrease in viral rebound. When you stop viral therapy, you know, if you don't treat them, the virus come back right away. But if you treat the viral particle, one treatment, you can reduce that viral rebound about 100 to 1,000 times. So then if it's a one monkey in here, the KC50 was treated for 2 to 3 times with this particle, that is self-controlled virus for about 2 years. Is without any treatment. It's usually this monkey is supposed to die. Anyway, so it was self-controlled. Then we look at what happened in this monkey. We find, you know, there's a robust production of neutralizing antibody. Right, so it's correlated with the viral control. So in summary, so the system we developed can directly kill HIV positive cells. Secondly, it also can stimulate immunity, generate antibody to block viruses as well. So it's a combination of all these different ways to control virus. So we're really want to move this into a phase 1 clinical trial. Is essentially in the future, it essentially give patients these particles. You know, in the HIV patient, it injects some of this HIV-like particle into the body. Hopefully, they can control for a few years without any treatment. Right? You maybe just do like every 2 or 3 years, do one injection. Very much like vaccine, but they're therapeutic vaccine. You can control infection. Okay, I'm gonna stop here. This is my lab member. Brian is the one. Is a major driver for the project. The monkey trial was done at Dulan Clinical Center. Thank you.
Okay, thank you. Thank you, Professor Wu, for the nice, wonderful presentation. I can imagine definitely this is very important research and brings hope to all the HIV patients. Thanks. I think we'll keep the questions to the end. Now, last but not least, it's my great honor to introduce our next speaker, Dr. Pengfei Song. Dr. Song is a highly respected expert in clinical pharmacology and regulatory strategy. Currently, he is a vice president of technical regulatory consulting at Parexel International. He guides groups in navigating complex drug approval process. Before joining Parexel, Dr. Song was global head of clinical pharmacology at IMAB Biopharma and spent over a decade as a clinical pharmacology reviewer and team leader, assessing hundreds of oncology drug applications for the FDA. He began his career as a clinical pharmacologist at Accio and earned his PhD at the University of Tennessee. Dr. Song is known for his deep expertise and leadership. Now let's welcome Dr. Song for his presentation. Thank you.
Thank you everyone. So I'm honored to be here with you along with these famous professors. Compared to other great findings, which are the core and foundation of any drug development, I will focus on more about the clinical development of the drug. And when we really get to the clinical development, we need to really interact with FDA, with EMA, because the human the drug development in human is a really highly regulated area. So how to work with the FDA, EMA come to the big concern for all the sponsors, particularly with the great development in Asia, particularly in China. People always ask, the clinical trial I generated may not be accepted by FDA. Would you tell me how I can do better to get accepted? So today I will just focus on this critical question. I give you my understanding about it. So here I give you a very interesting paradigm about the data, information, knowledge, wisdom. Generally, we were trained as scientists. However, we do not differentiate them. That is so important for our life, our understanding about the science, about the cognition, so important. However, I'd like to say in this setting, in today's topic, in this setting, I'd like to say, they are highly relevant data, just as we believe, fragmented piece of fact. And then information, we connect data to derive meaningful information. However, these information are maybe wrong, maybe right, maybe trivial, maybe important. We need to organize them into knowledge. And then we apply the knowledge as the wisdom to resolve the issues, to cure patients. How this is related to drug development. When we submit the drug information, data to FDA at the ECTD during IND stage or NDA stage, they are corresponding to each other very closely. We get data information as the CSR, TFLs data set, and then we need to put them as summaries, overviews, and then even the reviews, presentation to FDA as the knowledge level. And the next level, the top level, which is more critical, that's drug approval, and then approval letter, and then the label, drug label. So with this framework, get our mind, we will get a very clear idea what level or what we are working on is at. And then that will help us, with all kind of career or in the corporate level, what we are working on. And then with this data, information, knowledge, wisdom paradigm, we will work much better with a clear mind. When we get to the clinical part of the drug development, basically we wanted to ensure the favorable efficacy safety with 3 key deliverables: target validation, disease selection, dose optimization. These 3 key deliverables entangled together, and then that make highly risky decision making for the regulator to make, whether this makes sense or not. This entangled together. We always, FDA, as FDA reviewer myself, we always consider the regulatory risk, whether this causes patient's death or toxicity, or whether the benefit outweigh the safety. A lot of considerations. We need to tell compelling story to convince the regulator about our proposed clinical trial. How to do that? Today I'd like to say, FDA, regarding the foreign data, the trial conducted other than US, like in Asia, whether FDA accepts that. The answer is yes, FDA accepts that. However, with some considerations. The consideration is FDA has a principle at the law level, CFR level, at the guideline level, ICH guideline, also FDA guidance level, several levels, all state firmly foreign data is acceptable. with some conditions. Is first GCP compliant. The second one with clinical investigator with recognized competency, and then better with some global trial experience. And then the data set should be accessible for inspection by FDA. Even it does not really happen that often during IND stage. And then the core review issue is about the right side, that intrinsic factors or extrinsic factors a lot. So for different regions, that we need to the regulator needed to make sure the data you created at one region can be extrapolated to other region. So I think that a lot of factors. How we address that? We get two guidelines. One is E5, ICH E5, to handle the most of the intrinsic extrinsic factors, except the clinical practice as highlighted in that red box. That red box is generally is handled by E17, multiple regional clinical trial. IND during IND stage, because the regulators tend to accept the higher regulatory risk. So clinical pharmacology approach to adequately address E5 will mitigate the risk to make your proposal accepted by FDA. And sometimes, for the NDA, we have to adequately address the endpoint, the clinical medical practice as in the red box. EMA also followed very closely with FDA, follow the E5, E17 closely. They have some special considerations because they get the centralized, the decentralized, the different nations have different rules. We need to get that familiar with that. Also, recently they get the centralized joint clinical assessment, JCA, is about the payers kind of radio in parallel with the scientific radio. I think that's another important consideration. Basically, EMA follow the EU standard on GMP, GCP, GLP. And then overall, FDA, EMA accept the foreign data. I give you several examples, very well known, all based China related company, Chinese company. First of all, is about the toripalimab, Junshi product, PD-1 inhibitor. This one, I'd like to say it was approved both in US and in EU, based on 100% Chinese data. Why that was accepted? Because the prevalence of the disease, that one at the NPC, OSCT occur most in Asia, in China. And it also get a low prevalence in EU, in US, that make it unmet medical need. And then with the large phase 3 trial results in China, very well conducted trial. So EU and FDA all accepted, and 100% of Asian data. And then the second one was also famous, is sintilimab by Innovent. We got a ODAC discussion about this one. This one was not accepted. Also 100% Asian data. However, the clinical endpoint, also medical practice, will not, not that translated, extrapolated to US. They are different. And then this one, FDA recommend multiple region control trial for approval. Another one is uncertainty, is by Beta firm. They get the ALK inhibitor, and then it was approved. With some differences in the clinical practice, but it was approved with limited with restricted indication for patients who received who have not received the second line, third line ALK inhibitor. And that was a very successful case. One is BeiGene's example, zanubrutinib, even with 75% of data in Asians, and then only 25% data in Caucasian in US. That was also accepted based on very impressive outcome, and then no difference between the races. And another one from HUTCHMED, sorafenib, again is endpoint is not accepted. Also during the pandemic, the clinical data and the clinical side was not acceptable for inspection. That caused the rejection by both FDA and the EMA. So basically, with these several examples, we at Paraxo, we generalize the procedure for developing the drugs acceptable by FDA and EMA. If we have our existing data, the lower one, submit marketing registration application with the existing data, like a toripalimab model with all 100% Asian data, and then still get approved. That's one very shortcut, but not a norm, is an exception. If we develop the drug globally from the beginning, we need to take a seamless path to go global, like get the small trial, the dose escalation, backfill in Asia, and then get some early efficacy signal. You feel confident, and then come FDA for the IND, and then develop it globally. And then why some foreign clinical trial may be rejected? Okay, because issues again with regard to the general liability, applicability to the US EU population. As that's the most common issues with the drug. Another one is inadequately addressed ICH E5 ethical factors. There are some risks, nobody really feels sure about it. And then, but during IND stage, the regulator tended to accept the higher risk. And then it's still okay. We can work with the client to adequately address. The third one is the poorly conducted trial regarding the data quality, design, GCP compliance. I think that's the foundation. We need to make sure everything is correctly designed and conducted. Most common reason for the foreign data rejection is this one. Basically, is not well analyzed, well presented, and then we have a lot to say, but we cannot speak it. So basically, we need to conduct integrated dose exposure response analysis to build a knowledge system for decision making. FDA like to see compelling story rather than fragmented data or information. So take home message: FDA accepts foreign clinical trial data, comply with the ICH E5 regarding the ethical factors and E17 MRCT for the generalizability and the application of the clinical trial. And then FDA expected strongly demonstrate the knowledge rather than information or data level, regarding the disease, regarding the mechanism, regarding the dose. Also, the FDA EMA really like you to engage them early on to make sure we get everything on the right track at the beginning. Thank you so much. That's my talk. Thank you.
Thank you, Dr. Song, for the wonderful presentation. Definitely very important topic. I think later on we'll have a question from the audience. Hopefully we have a deep communication. Now, before we lead to the Q&A session, let me present a certificate on behalf of the meeting organizing committee to express our sincere thanks to all the speakers for your outstanding service to this science community. Let's take a picture over there before the sign. See that the certificate is make sure let's stop the song. Let's stop the wood. This is for Epsilon, and this is for. Okay, let's take a picture of the. You can go with us. And you can show your certificate to the audience and hold it together.
Really comfortable with that. Yes, no light on the face. Yes, that's
Cool. Okay. 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 Thank you. Okay, now it's our Q&A session. We have talked about 10 minutes. We have a break for we only have minutes off. Sorry, there's no time for break then. And because we need to join the main conference room for the ending remarks. So we only have 10 minutes, right? So let's take some questions from the on-site and then we'll go to online. So any questions for the speakers for this session? You can raise your hand. We can hand the microphone to you. Looks like the audience needs some. Okay.
Yeah. Actually, very interesting topic by the other presenter. Yeah. Can I ask some question about the Dr. Wu? Yeah. Very nice the talk about the reef target vector development. So can I ask the first question is that have you observed some the toxicity for this reef treatment vector in vivo study?
This is a very good question. So actually, there one set of data I did not present here. We used another vector called trap6, right?
Yes.
So that vector actually their toxicity actually triggered the death of 2 monkeys. So that and now we couldn't use that vector. So now I think the herpes TK vector the fairly safe. We don't go off the medicine, right? So they're largely very well tolerated. The Pep6 vector was toxic. We know it was, so we abandoned that vector. Then later, I did not show that data.
Okay, sounds great. So can I ask another question? Is that how about the title for the vector for the treatment? Because that's important correlated with the manufacturing or something. Yeah.
So we essentially was highly concentrated.
Okay.
This is a particle. I have to tell you because this is the global concept study, but not really industry standard.
Okay.
So roughly concentrated about 500 folds, right?
Okay.
So we do not have if you not remember, we have genome copy number. We really because those are the toxic genes, so we don't have a titer.
Okay. Right. Oh, so the last question is that we're maybe very naive. So can we use the LNP instead of the vector for your treatment? Yeah, lipid nanoparticle.
So, yeah, but then I think the particle has very much the same structure like the HIV binding to the receptor. Enter the same cells. If you use LNP, it launches into many cells.
Okay.
So I don't know whether it's going to be better or worse.
Yes.
It's something we certainly want to try. But I bet the particle probably better than the LNP because we select for HIV particle cells.
Okay. Okay. Thank you. Thank you so much.
Thank you. Any more questions from the audience?
So what you make
work? And you remind it later or then
Right.
why didn't it work?
So why the shotgun two,
Yeah.
the approach did not work, right? Okay, so first I don't know, right? Because I don't try them, I don't know. But the other people try them, they were not able to show first. Immune system is always a very delicate balance, right? When you shock the immune system, so what is the how do you shock them? You shock little, it does not active virus. You shock too much, you kill people, right? So where is the line? Right, you can shock. Secondly, even you can shock, it doesn't lead into kill.
Yeah,
Right.
in my mind, though, the shock and kill.
Right.
So say for instance, you sequence when you have a sequence, right? Can we use gene editing?
Right.
Table?
Yes.
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 Yeah.
For moving the human die.
Right. So you there are people try to use CRISPR-Cas with editing HIV. The issue you can do them in test tube. There's no issue. But once in the body, where to find the cell? Right. You can't find every cell to edit in. So that's the question. Right. So there are many cells, and we even don't know where they are. How you deliver your editing tool into those cells, right? So those are the issue.
Thank you.
I have a question for Dr. Song. So regarding accepting foreign data, I hear that it seems that FDA have no issue for phase 1 trial that completely done like in China. I wonder if that's accurate, like for cancer studies.
Actually, generally, yes. Answer is yes. So if you present it very well with the analysis to see the trend and make good use good use of the findings in Chinese patient population, and then you make an argument about the safety efficacy you identified in Chinese population is applicable here. So, however, some sponsors did have the issue. When they really presented the clinical finding in the Chinese population with kind of without a good data analysis and the good presentation, FDA team feel really concerned about the data in China. They very at the extreme situation, FDA asked them to do the dose escalation again. That's not acceptable because the data in human is really precious. We cannot ignore it. And then FDA would not ignore it at all. But unfortunately, sometimes, when the data does not really convince FDA about that, they start from the animal data, animal-based starting dose. That's not right. FDA should not do that. However, unfortunately, the data was not well analyzed. Not many times just the data done, and then FDA I have no confidence at all. So you need to restart everything from scratch. I think that occur. That did occur, in the past, but not that common. Yeah, good question. Yes.
Okay, just a follow-up question. So some study that they so phase 1 study in China, they may not reach to like before they run the study, reach out to the FDA like a reviewer or the project manager, because at the time they might not have a team here in they don't know how to reach out to FDA, and maybe after the phase 1 they feel like they have confident maybe get to the US market. Maybe at that point it's too late to discuss with the FDA. Do you have any suggestion?
Not at all. Not at all. Anytime the sponsor can reach FDA, even with the phase 3 data, without any active IND, they may still come to FDA to ask for NDA submission. Then there is no restrictions on the stage of the drug development. No need to worry about it. Anytime, with the very well analyzed data, with clinic, well conducted clinical trial, you can work with the FDA to figure out what's the next step. Anytime, FDA is very open to all the good drugs, particularly in China. So many good drugs come to US. FDA is open, very open to those wonderful drugs.
Thank you.
Thank you.
Xinquan, do we have any online questions? No. Okay. If no question from the audience now, I have a quick question for Dr. Han. Oh yeah, very nice talk. I know your presentation or your research definitely is very important. The disease model is our hope for further deliver cure for the patients. Could you hear a successful story where Drosophila model aided treatment development? Thank you.
Yeah, so great question. So Drosophila has been used to discover many fundamental principles in biology and genetics. However, it's only recently the Drosophila model has been used in larger scale for disease modeling and for developing treatment. There are very few examples, but there are some examples, and we are aiming to expand those. So one example I mentioned in my talk is identification of the coenzyme Q10 as the treatment for COQ pathway mutation induced kidney disease. So more and more patients, particularly for the pediatric patient with kidney failure, classified as nephrotic syndrome, and more such patients are being sequenced right now, because the idea is if their disease is caused by the COQ pathway. So now we already found 3 genes: the COQ2, COQ6, and COQ8. So if the variants is identified in any of these 3 genes or even more, like there are about 12 genes in the entire pathway. If we found the variant among those genes, among those 12 genes, then we have a treatment already. Because after the study from my lab using the fly model identify coenzyme Q10, their mouse model has been done, and then there's a patient trial has been done. So this is one of the great examples, starting using fly and then using mice and then using develop treatment for the patient. And my colleague Ross Kagan from the Mount Sinai in New York has used fly to develop a cancer treatment drug using a large scale genetic screen, and that drug is being tested in patient right now. And there are also neurological disease drug being tested in Baylor from Dr. Hugo Ballin's lab, which probably soon gonna be in clinical trial. Okay.
Thank you so much, Dr. Han. I asked for one story. You actually shared 3. That's very exciting. I think for beings online and maybe I think you have another question for Professor Wu, right? Before that, let me call for questions again. Don't be shy and don't feel that any question is a good question. No bad questions at all. So last chance to ask a question direct to the real expert in this area. So maybe next, it's your turn to ask.
Yeah, I'm just thinking like, what do you think about the future of this HIV therapy, right? But particularly for the HIV associated, like, probably kidney disease.
Yes, kidney disease. First, I think in HIV, if you think about completely wipe out the HIV, you really absolute cure. You clear the reservoir 100% from your body. I think is that's bar is very high. It's really difficult to achieve, right? So you clear anything from our body is difficult, right? So I think in the future, likely their immunotherapy, right, could be play a huge role. Like you have broad neutralizing antibody, give to patient, right? Also, it's not like a permanent. You likely require every few months. If the virus mutated, if it's keep mutating, they have to generate cocktails of neutralizing antibody, give to patient. So also our approach essentially inject the vector. It mutated with the virus, right? So I don't know whether it's gonna be better than the broad neutralizing antibody. Hopefully, I think our approach could by the end is better than anybody. Directly injecting the antibody because it's mutated with the virus. It's a past HIV. It hopefully can last, few more years, right? It can just very much turn the patient into the long-term non-progressor. Many years was already treatment. So that's our hope. For kidney disease, I really don't know. I need knows very little about HIV. I know there's an issue, but I really don't know how to treat them or what is the mechanism.
It looks like there's a field there called HIV nephropathy. A lot of nephrologists are trying to develop treatment. Right now, it doesn't seem to have an effective treatment yet.
Right. I think your fly could be a very beautiful model. Maybe I can collaborate with you to study them, right? Yeah, especially also the antiviral drug, how they impact the kidney function.
Yeah.
Right.
Yeah.
Yep. Thanks, Professor Wu. Do you have a question for? Okay. Can you please hand the microphone?
This may sound very speculative, but I was wondering, to what extent now with all the AI technologies that is coming out, right?
Right.
Can they focus on using materials, the artificial materials, right? Based on the content, the knowledge that you know about the real things, right?
Yes.
And then apply it back.
Right.
Rather than using animals,
Right. I think you're a scientist. Looks like you're a good scientist. Right. The question you're right on the point. Exactly. There are scientists doing use AI to predict the mutations of the envelope, the HIV, to predict where use AI to mimic how they mutate in the future. Then they design broad neutralizing antibody, so they can even the virus is not there, the antibody is produced, right, for the future. I think they're just wonderful. You should do research, right? Yeah.
Thanks for the nice answer. I have a quick question for Professor Wang. Could you please maybe just a brief words to like prospect, like what do you think your research field will go? Thanks.
So I think most of my works focus on the drug safety and also involve the developer like in vitro this NAM related models to better evaluate this process. But I think it's truly a combination, and I think like one area I think right now FDA doesn't have a clear regulations like how to standardize all these new NAMs and everything. And so we actually collaborate with FDA on several projects to evaluate a number of these commercial NAM models, but like where's the criteria and all those things. And so regarding this project for the drug development, and we are continue to optimize the analog of this class of compound and improve the in vivo study. Currently we're working on that. Yeah, let's just stop here.
Great. Thanks. Many thanks. Let's give another round of applause to all the speakers. Thanks for the wonderful job. Thank you. Now let's move, or we should move to the main conference room for the any remarks.
Thank you.